For those of you who don't know me, my name is Tasman. I'm in the College of Science and Engineering in Zoology and Ecology. And it gives me great pleasure today to welcome Carsten Schradin and Lindalani. Is that how we say that one? Lindalani and Carsten. Let's just be a little bit informal today. I've known Carsten for a very, very, very long time. We've established that it was now 2001 when we first met, which is longer than some people have been born or been alive. So that makes me terrified. But it's great. So Carsten actually was in South Africa, which is where I'm from, and he was studying a postdoc looking at paternal care in African striped mice. And so he's a behavioural ecologist by nature, very interested in social systems. And he's going to be talking to you today about a really cool system. I'm not biased at all. I studied the same system for my PhD, so no bias whatsoever. But his focus has been largely on social flexibility and also this concept of harshness, because these animals come from a desert environment. And then enter Lindalani. Now, I haven't known Lindalani that long. I had the pleasure of meeting at a conference, I think in 2023, 2024. And Lindalani also focuses on sociality, but with a little bit of a twist. So she did her PhD on bush karoo rats. Now, bush karoo rats are also fantastic little rodents, and they love to compete with striped mice. But they're solitary, or should we ask the question, really are they? Okay, so they approach sociality from slightly different perspectives, but both very interesting perspectives. And I hope you'll learn a lot today about sociality in a group of animals which I love, which are rodents, and I will stop talking and pass them over to you. So we'll start with Carsten, and then Carsten will pass over to Lindalani. Thank you.
Yes, good afternoon. Thank you everyone for coming here for our presentations.
Sorry, Carsten, can you try not to open it? Just because of that.
Okay. No, it's okay. I can do it like that. So it's a pleasure to be here. I'm going to talk about one of the two main aspects of my research on striped mice. It's adaptation to droughts. It's also, I think, of importance for ecosystems in Australia, though Cairns is the worst place to talk about it here in the wet tropics. It's more related to what similarities you will find out in the dry lands of the outback. But as it's only 1-2 hours drive from here, I still think all of you are familiar with this and can see the similarities. So climate change and human-induced accelerated climate change is a phenomenon we're all familiar with, and one of the consequences of climate change will be the droughts, which naturally occur in ecosystems, will occur more often, will become more severe in the future. And this has often then been related to being bad for animals and related to the concept of harshness. So harshness is an important concept in ecology, with harsh environments often described having low rainfall and variable climate that favor the evolution of complex animal societies, for example, with cooperative breeding. And increasing harshness due to climate change, such as extended periods of heat and drought, has been identified as a major threat to biodiversity and ecosystem functioning. So harshness is important both from the point of view of social evolution, but also of biodiversity and conservation. However, though this term harshness has been used in multiple publications in all of these fields, it has never been properly defined. And I think in science, it's very important that we define, that we know what it actually is that we are talking about. And wrongly, often this has been assumed to be just the same as stress or long-term stress. So everything that's bad for an animal is harsh, is stress, is very bad. And here I want to argue that it's important to differentiate between stress and harshness, and that these are two fundamentally different problems animals are facing, and they're fundamentally different with how they respond in their physiology and in their behavior to these problems. So that's what I just said. Here I will argue why it's important to differentiate between stress and harshness, both for understanding ecological and evolutionary processes and for nature conservation. So First of all, what is stress? This is already something that has been debated for many decades. And when I was a PhD student and measuring corticosterone and cortisol, the stress hormones, there was already a big debate what is stress. And the best definition I ever came up to, at least for vertebrates like mammals, is stress is what activates the physiological stress response. So the stress system in vertebrates consists of two different systems. It's first the sympathetic nervous system, secreting adrenaline, leading to an increase of the sympathetic nervous system, increasing your heartbeat within seconds. And second, the HPA axis, the hypothalamus pituitary adrenal axis, with glucocorticoids like cortisol or corticosterone, depending on whether you're a primate, then it's cortisol, and you're a rodent, it's corticosterone. And most people measure in the field cortisol or corticosterone because these hormones are very stable, can be easily collected in blood samples, where adrenaline is a hormone that breaks down very quickly, and it's very difficult, if not impossible, to get samples to measure this immediate stress response. So for me, stress is what activates the physiological stress response in vertebrates, both the sympathicus nervous system and the HPA axis, which means for other taxa, when we look at insects, when we look at plants, what I'm talking here about might not apply directly, because it's also often discussed in plants. They have water stress, heat stress, but obviously they have a different physiological stress response, so the pattern here might be different. So both stress and harshness challenge survival and reproduction of individuals, which is why these terms are often confounded and used synonymously. Stress, these are detrimental environmental events activating the physiological stress response. The stress response leads to the activation of energy resources. So everything the stress response does is to give more energy to the animal so it can do something against the stressor, which is typically to fight it or to run away from it. And with this, the animal overcomes the stressor. Harshness, in contrast, are long-lasting environmental conditions which decrease energy availability, because there's less food in the environment. And the harshness response must be to save energy. If there's little energy in the environment, you should not activate the stress response and spend more energy, but you have to save it. So for harshness, the strategy is to endure it until it is over. Examples for stress are predation, territorial encounters, a new dominant hierarchy, think of a group of baboons or other primates, or bad weather when there is a cold front coming in and the individual has to activate energy to survive the cold night till the next morning. Harshness examples are low food availability, low water availability, cold, winter, and this lasts for months. For example, winter in the northern hemisphere in Europe, when animals have to go through several months of low food availability and the cold, or heat, which is in summer and also can be for several months. Think about the outback here in Australia or the deserts in Namibia and South Africa. And here the harshness response is to save energy, to endure it until it's over. To show this in more detail, we published this paper in trees some years ago where we have the physiological stress response. Here we see the adrenaline levels increase within seconds after the stress occurred, which is shown here. Okay, now they go too fast already. No, which is shown here. Period 0 is when the stress occurs. Adrenaline goes up very quickly, and also declines quickly. Corticosterone or cortisol, in the mouse it's corticosterone, increases slowly between 3 to 5 minutes, and gets a peak, and then decreases again. Long-term stress or chronic stress is when the stressor does not disappear, and thus the adrenaline and the corticosterone levels stay increased over longer periods. Here before we had minutes, and then if it doesn't disappear, this occurs over the period of days. And long-term stress, then finally the stressors overcome. Maybe this is a tupaya, a tree shrew that has been studied by Van Holtz in captivity, that are typically living in pairs. And when you put a second male into the pair, the second male is extremely stressed. And under natural conditions, what would happen is after some periods, the second male would disperse and go away from the stress and would go down again. If this does not happen, because you keep them in captivity, you come to chronic stress, which obviously is detrimental for the health, and this you find normally only under captive conditions in animals, and the only free living animal I know that shows chronic stress are humans, because they are in a workplace or they are in a relationship which they don't escape, and they're there for days and weeks and months and years. But in animals, normally under natural conditions, you won't find chronic stress. Now, harshness is very different. Here we don't talk about minutes or days, but about weeks, and it comes continuously, and then the stress hormone levels do not increase, but actually the hormone levels of the HPA axis, corticosterone, decrease continuously to reduce energy expenditure. These so-called stress hormones, corticosterone, cortisol, so the catecholamines, they're also important for basic metabolism and provide energy throughout the day. For example, in the morning, before you wake up, your cortisol levels increase, so you have more energy and you can start your daily activities. So this is what we summarized in this tree paper, that we have differences. Normal stress, this is something within minutes. Long-term and chronic stress, we talk about days. But harshness is something that happens during weeks, and the physiological response is very different. Here we have an increase of stress hormone levels, and here we have a decrease. So how will climate change influence this in the future for stress? It's mainly bad weather, heat waves, floods, extreme colds, where there's bad weather conditions for one or few days will increase the frequency in the future. But for harshness, low food availability, low water availability, cold and heat are things that might, due to climate change, increase tremendously. The frequency of droughts and the duration of droughts might increase in the future, for example. And this we've been studying in the example of the African striped mouse, which we study here in the Guchab Nature Reserve, which is 600 kilometers north of Cape Town. If you drive up there in the so-called Succulent Karoo semi-desert, it's a winter rainfall area where we have rainfall only during winter, and then we have no grasses there because in winter it's too cold for the grasses to grow. This is why we do have succulents, and there's nearly no trees in this environment. So it's quite seasonal. In winter, there's relatively high food availability because there's always some rain, and then there's plants growing. The resolution is not so good. Everything that's dark here, it's green or orange. It's all food plants, short-living food plants that are growing for short periods, and everything of this is striped mouse food. But in summer, all of this is gone. There's mainly sand and dead leaves left, and there's very little food availability, and temperatures also increase dramatically. And we have been studying now for 25 years, this system. Here you see a photo of our excursion from the Wits University, including Tasmin in 2005, when they came to visit this research station. And I show here very quickly an overview of different responses of the striped mouse to harshness we have been found. Also with Tasmin, we wrote a paper in a review paper about resilience to droughts in mammals, where we summarize all the physiological and behavioral responses of animals to the three main problems of droughts, which is low food availability, heat, and low water availability, if you want to read this up. And in this paper, we still called all of this stress. And in the meantime, I changed my mind and I call this harshness because it lasts for a long period of time. So we have first example how striped mice deal with this low food availability. You see here a time-lapse recording during the night. It's a diurnal species active during the day, and here's a striped mouse nest, and they sleep closely huddled together, warming each other during the night. This is expenditure measure much more energy than the mice that are sleeping in hunting groups, except when they reach communal quality or characteristics. So they can save up to 25% of daily energy expenditure. We measure this with daily W level water, which is a huge amount of energy saving for this desert living rodent. And I think this is the main reason why they do live actually in groups. Because by sleeping together, warming each other at night, and it can get quite cold in the desert, they save a lot of energy. What they also do then, when they get up, they get in front of the nest and they are sun-basking. You see here a mouse with pillow erection, and you might see here the striped mouse is one of very few mammal species. The polar bear is another one which does have black skin, and they sunbask in the morning, show pillar erection such that the sun rays go directly on the skin, warming up the animals passively. And here also with respirometry, we showed that as soon as we put shade over the mouse, independent of temperature, the energy consumption increased dramatically. This here was before the sun was on the mouse, because it was still in the morning, the sunrise, there's decrease of energy expenditure, we shadow the mouse, and the energy expenditure is increasing again. These were studies done together with Mike Canterbury, who is at Queen's University in Belfast in the meantime. Then several studies have been done by our postdoc Rebecca Rimbach, who is now at the University of Münster in Germany. And first, she showed by following mice for the entire activity period during the day. These mice come out of the shrub, they're diurnal, it's an open habitat, a student observes them and every minute writes down what the mouse is doing, but only for 6 hours, then the second student takes over, and like this we could follow mice for the entire 12-hour activity period in the field. And what you see here is overall activity in the dry season, overall inactivity, so that's resting. In the dry season, there are both the females and the males are resting significantly more than in the moist season when there's lots of food available, and they also spend less time foraging. Why less time foraging when there's less food? The only food they have, the main food, are these shrubs. These are Psychophyllum shrubs, and there are emergency food resources always available, but of low quality. So they can eat this, but not much more, and there's nothing else. It doesn't make sense to run around and look for something that does not exist. But in the moist breeding season, they can walk around here and find little plants, little flowers that are high in protein and are quite nutritious. Here, maybe I should put this in future as the first result, because it's the main difference to stress. These are corticosterone levels in 4 different social categories of striped mice: breeding males, breeding females, philopathic non-breeding females, and philopathic non-breeding males. And during the breeding season, in all except the males, these corticosterone levels are much higher than the non-breeding season. And like I said before, corticosterone is not only a stress hormone. We talk about basal levels. They're also important to give energy, for example, for females, for lactation, and for pregnancy. The reason the breeding males are the only ones that are different is because too high corticosterone levels are not good for spermatogenesis, and during the breeding season, spermatogenesis is quite important. We find it for many other hormones, which all of them also have some metabolic function, like testosterone decreases in all categories during the non-breeding season. Prolactin, that is related to reproduction in mammals, also decreases during the non-breeding season. And thyroxine, that is a very metabolic hormone, is directly related to the availability of food plants in the dry season. With low food availability, the levels of T3, triiodethionine, are significantly lower than during the food-rich breeding season. Interestingly also, they show an impact on growth. Here we have the body length of both females and males as a period of their age. And the striped mice, normally, a small rodent of their size would stop growth when they're about 2 months old. But striped mice continue growing even within the dry season, but at a very slow rate. Body size is very important for fitness. Larger females have larger litters. Larger males get access to more females in the coming breeding season, but they are growing for a very slow amount during the dry season. Another interesting result we found is that there are seasonal differences in the size of different internal organs, and the one I want to emphasize here is the brain, that actually in the dry season, the food-restricted dry season, the brains are relatively smaller to the rest of the body than in the moist breeding season. That's called the Daniel effect, has been first described in shrews, which show shrinking brains in winter in Europe. And the brain obviously is one of the energetically most costly organs of mammals. And so they can reduce the brain size to save energy. How they do it and what is actually decreasing, we do not know. But it is the first time that this has been shown in a mammal from the southern hemisphere, which has been described for shrews in the northern hemisphere for many decades. They're also decreasing their resting metabolic rate in the dry season by nearly 30% compared to the breeding season. And this is also related to food availability. When this here is shown in the moist season, the line, when there's a lot of food, the food availability has no influence on resting metabolic rate at all. They just all have the optimal high metabolic rate. But in the dry season, when food availability differs between years and months, there's a significant relationship. The harsher the dry season, the more they reduce the resting metabolic rate. And all of this then also impacts the total daily energy expenditure as measured with the doubly labeled water method. They can reduce the energy expenditure by up to 40% in the dry season compared to the moist season, both by their flexibility in their behavior and the activity patterns and the different physiological mechanisms I have shown you. So the harshness response of striped mice consists of multiple mechanisms like social thermoregulation in huddling groups, basking to passively warm up, reduced activity, reduced levels of different metabolic hormones, reduced growth rate, reduced brain size, reduced metabolic weight, and all of these mechanisms together lead to a reduction of 30%, in the other it was even 40%, in daily energy consumption, which is a significant reduction so that they can endure and survive the dry season to breed in the coming breeding season. And these results have been summarized together in this review in the Journal of Zoology that just came out at the beginning of this year. So to come to Australia, I think this harshness concept might also be important for Australian fauna, especially in the outback. For example, kangaroos have evolved this very energetic, efficient way of going through the outback of locomotion. They spend only half as much energy for movement compared to an African gazelle. The zebra finch, you might be very familiar with, is also like the striped mouse, spending the night together in sleep nests, also outside the breeding season, where the birds warm each other up and save considerable energy. And the field lizard also reduces, like the striped mouse, its metabolism by 25% in the dry season and activity to save energy when food is rare. Whether this harshness concept is also of any importance in this area here in the wet tropics, I'm not sure. I do not know whether the monsoon season is a season of extended period of harshness or whether it's rather one cyclone coming in and it's more stress of a few days. That's something you could discuss between each other. So to conclude, harshness is not stress. Stress leads to an active stress response that increases energy expenditure to overcome the stress. Like harshness leads to activates the harshness response, including a reduction of stress hormones and reducing energy expenditure to endure the harshness. Striped mice have a specific harshness response that is different from the stress response. And the harshness concept should also be applied to other species and to conservation. For example, in harsh environments, to have additional stress on the animals, for example, by tourism, by activities of humans, like building, might make the survival of the individual even more difficult. Thank you very much for your attention. And with this, I will be handing over to Lindelani, and then afterwards we can both receive questions, and Lindelani will talk about the evolutionary benefits of solitary living, and there's many solitary living species here in the wet tropics, and that might be also of interest for you. Thank you.
So thank you for the introduction. So my name is Lindelani Makoya, and I'll just start by giving a brief outline of who I am and where I come from. So I studied my BSc, so my Bachelor of Science at the University of Johannesburg, where I was looking at doing a major in both botany and zoology. I then moved on to the University of Pretoria, where I studied the biogeography of endemic rodents, so the endemic mice and rats, and as well as also the invasive rats that occur within the country. And during 2020, at a time when we're all supposed to be socially distancing, I was given an opportunity to study the bush karoo rat, which is a solitary living species, so something which was a bit of a benefit for this rat to be spending time on its own. And from this, I was then first hired as the research manager for the Succulent Karoo research station. And then I started already collecting data for my PhD, which I did with the University of the Witwatersrand, as well as the CNRS, which is a research institute in Strasbourg, France. And currently I'm a postdoc, continuing with the field data in South Africa and still being supported by the CNRS in France. So the main reason why I was interested to study solitary living is that we wanted to create a community for all the studies involved or related to the ecology, the evolution, as well as mechanisms of solitary living. And this is because we don't understand really solitary living if we cannot understand its alternative, which is pair and group living. So we need to first start by understanding why individuals live solitarily. And this is because a fundamental problem in social evolution is that this misconception that solitary living needs no explanation. And because for many decades, behavioral ecologists have just studied only why animals live in groups and neglecting why individuals are also living living solitarily. So, which is why we came apart with this study, as well as the website where we bringing together everyone to study solitary living. So I based my definitions from Peter Capella's work, which looks at the social system, which is a conceptual framework that looks at defining 4 components of the social system. So we have the social organization, which looks at the group composition of the adults, the group size, the kinship patterns, as well as the spatial structure. We then have the social structure, which looks at the social interactions both in and within groups, the social networks, and communication. We look at the mating system, who mates with whom, who reproduces, as well as alternative reproductive tactics, and then the result of that, the care system, who takes care of the young. And all of this is all interrelated, and each component informs the other. And we see that within each component, we can have 6 different types of categories arising. So within the social organization, we can have solitary, pairs, multiple males, multiple females. So over 6 different types of categories can arise from the social organization. And when we look at the social structure, we see that we can have either individuals that have a pair bond or not, whether they are territorial or not, whether there's some level of dominance hierarchies, as well as whether they show cooperation or not. And from this, we can have a combination of over 8 different types arising from it. With the mating system, we see that there are over 4 different types of mating systems possible, from monogamous, polygynandrous, polygynous, as well as polygynandrous. And in the care system, we have all the 6 different types of care systems possible, from no care, maternal care, biparental care, communal care, etc. And when we look then across all of them, we see that there's over 1,000 different types of social systems that can be categorized. And this solitary living is just one component of this different social system. So for example, when we look at the European hamster, this is a prime solitary species that has been described, and its social organization is that it is solitary living. Its social structure is that it is very territorial. Its care system is that it shows maternal care, and its mating system is polygynandrous. So this is just one example of a type of solitary species. But first, we need to define what I mean by solitary living. So we, on our review, we define it based on the social organization. This just went off. So we define it based on the social organization of the social units, where both adult males and adult females sleep and forage alone for more than 50% of the time, and mainly mate for courtship and mating. So if we look at across the different taxa we have, we can see that solitary living is a very successful adaptation across the animal kingdom. So we have a solitary bee, which shows maternal care. We have the Siamese fighting fish, which shows paternal care. We have the common toad that doesn't seem to have a care system. And then we have the red-footed tortoise, which also doesn't really have a defined care system. We have the southern cassowary here in Queensland, which shows paternal care. and the Middle Eastern mole-rat, which shows maternal care. So first, these are all solitary species, but based on their care systems, we now having these 6 different types of categorization of the different solitary species. So in general, solitary living is a successful adaptation to many environments. So far, solitary living was only believed to occur in about 68% of mammal species. However, in our review, we saw that only about 22% of studied mammals are actually solitary living, with recent comparative studies showing that solitary living is often not ancestral. For example, in the Artiodactylus, which was published in 2020, in the elephant shoes, as well as in the primates. And we see that the general, the ancestor for example, for the primates, the ancestor was believed to mostly be solitary living, but we find that it was actually pair living, with the overall variability in its social organization being explained by solitary living. So we know, to understand this variability, we need to also include the solitary living individuals. We see also that solitary living is a successful adaptation for many environments. So, as Carsten mentioned, harsh environments are said to favor the evolution of complex societies with cooperative breeding. For example, in the Kalahari, which is a semi-arid desert in southern Africa, 6 of the 90 mammal species that were listed are cooperative breeders, while 30 species are solitary living. But also within the same environment, many social, but even more solitary species are adapted to it. So where could this adaptation or this success really come from? So we know that the main costs of solitary living arise from missing out on the benefits of group living. So for example, in particularly group living species, we know that help by close kin can help to increase its direct fitness, and also helping close kin can increase its inclusive fitness. So from that, we see that with the kin selection theory, kinship is important to understand social evolution, particularly for group living species, with species that show alloparental care, so help us at the nest, as well as for cooperation. But kinship might also be an important concept to understand for solitary species, whereby studies have shown that interactions with neighbors can lead to a spatial structure that could be characterized by kinship. So for example, in the giant kangaroo rats and in the mammoths and also in the leopards, they have found that individuals have this kin-based spatial structures whereby they interact more with their kin and they're more amicable with the kin, even though they still remain to be solitary living. So this brings me to my own study species. So I studied the bush karoo rat, which is a small diurnal rodent, about 100 grams. It is a central place forager, so it always has a central lodge from which it forages from, and it just moves out into the field for the foraging grounds and then comes back. It is endemic to the semi-arid regions of South Africa, and it occupies an open habitat, allowing us to be able to easily observe it in the field. What is very special about the rats is that they build these extensive stick lodges at the base of shrubs, and what has been reported and what we have also seeing is that multiple generations tend to maintain these lodges. So this is an example of the lodge, which is a dome-shaped structure, and these are just a bunch of interconnected sticks that have been put together by this rodent. And here you can see one of the rats still carrying a piece of stick towards this lodge, and this is just the size comparison to a juvenile from one season. So the stick lodges are just, like I mentioned, dome-shaped structures. You can see the dome shape here and all the sticks connected to it. And they're built at the base of shrubs. So this is still a stick lodge. You can't really see it, but it is. You can see more the shrub that it is built from. And once the stick lodge becomes quite big, it can overgrow the shrub itself. So, as mentioned earlier, the study site is the Succulent Karoo, which is Namaqualand, which is a biodiversity hotspot with this very distinct 2 seasons, so the moist spring season as well as the dry summer season. And this is just the rat's distributional range. Another reason that made us, or made me interested into studying the bush karoo rat is that previous populations had been studied in this region, and they were said to be more group living, with reports of multiple individuals being found within the lodges. However, our observations up here, we were finding that they were more solitary living. So, but with the studies down here, there were no published records. There were only student reports, anecdotal reports. So they were still missing the empirical data to really understand the social organization, which is where I then came in. So what I did was to look at the lodge surveys and measurements, just to look at signs of occupation for the lodges. From there, I then did trapping by placing traps on the entrances of the lodge. I then also did focal animal observations. The main thing I did was to attach mini GPS data loggers on the rats to study their movements, not only for individual home ranges, but also to see if there was also any overlap between the neighboring rats. I also did dyadic encounter and filled into the test just to see how individuals respond to other individuals within the neighboring community and whether or not they were kin or non-kin. So the first thing I found was that the bush karoo rat showed a kin-based spatial structure. So here, the green, each color represents a matriline. So we see that there was this matriline where a mother and her daughters were living in close proximity. There was also one other matriline down here with the one female slightly a bit out and another matriline here. And this is just our fill site, and each block represents an area, our own designated areas, and each white spot represents the lodges that we sample at. And here you can see much clearer with the kin-based spatial structure, not only in space, but also in time, where they seem to, they overlap whenever there's more overlap between the kin individuals than non-kin individuals. What I have also been interested to see is just this temporal overlap, because it's not only important to see whether or not individuals sharing a space, but are they sharing the same space at the same time, which is something I'm still trying to untangle. And as I mentioned, so not only did the individuals, so kin individuals lived much closer to each other, irrespective of the season, than did non-kin individuals. And in turn, they also had more overlap in their home ranges between kin than between non-kin ranges, just by virtue of them being living closer to each other. So for the solitary individuals, so that when we're looking at their social organization, they can also have this non-random individualized social relationships, which is their social structure. But so when we look at solitary living or solitary species, solitary living does not necessarily have to imply being asocial. One example of this is been recorded in the crocodiles, where they found that one individual, or beta again being solitary living, it also had this. So it also had a lot of interactions with other individuals within its range, which was non-random. So they seemed to be more common, this overlap as well as interactions between these individuals. The same was seen for the puma, where we found these individuals interacting more amicably, particularly at kill sites, and them forming this sort of social grouping with all the different colors where they overlap in their ranges, but also the number of interactions that they had was quite significant. So there are different mechanisms that could be involved to define solitary living. So they can be ranging from behavioral to physiological, and also this could lead them to us having this very highly aggressive solitary species, such as the European hamster, where active avoidance, because they actively avoid each other because they are aggressive to each other. And then we can also have the bush karoo rat, where they could be solitary living because maybe they just due to a bit of lack of bonding, or maybe just avoidance, not necessarily related to aggression. So again, solitary living being a part of two different social systems. So of the different over 1,000 different social systems, when you just look at solitary living, we can see that its social organization will be, of course, solitary living, where we can have a defined spatial kin structure. The social structure we can see that there could be non-random interactions, and these species that can be very tolerant to very highly aggressive. We can have a myriad of different care systems occurring and different mating systems also occurring. So solitary living is just one component of different social systems that needs to be studied. So in conclusion, solitary living is just a successful adaptation to many environments, and solitary species will have this kin-based spatial structure, which I believe warrant further investigation. And solitary individuals, from their social organization, have non-random individualized social relationships, so their social structure. And it is just one component of different social systems with different behavioral as well as physiological mechanisms underlying whether individuals range from highly aggressive to very tolerant species. And we really need to study solitary living as an evolved trait to understand social evolution. So I'd like to thank you all for listening, and I'll be happy to take your questions.
Question?
Now's the time for interrogation.
Thank you, Will.
Hello. It's about the first one, and I don't know if it's a completely stupid sort of question, but thinking about harshness is like a deficit model, right? So that it's a good time and they're reacting to a bad time. I wonder if the reverse is actually true, that they have a base level that they function on, that only when good times can they respond the opposite way. Is that a crazy, like it's a sort of different way of thinking about what harshness is. Harshness is the state or the base state, whereas the good times when you can upregulate all that stuff is the sort of favored response. Does that make sense to you?
I'm not entirely sure, but of course, without the good times, animals cannot reproduce and they would just go extinct and have zero fitness. So I think we cannot put the harsh time as the baseline, because the baseline must be somewhere where populations can be maintained and where individuals can succeed evolutionarily. So the harshness is really a period they have to endure to be able to maintain the population and any reproductive success. I'm not sure if I understood your question correctly. Sometimes for humans it might feel like you have very only harshness in your life, especially when you look at philosophy. In the history of philosophy, Buddhism and other religions, it's like there's only suffering in our life. But even there, I mean, humans also reproduce and succeed in harsh periods. They don't shut it down totally like many seasons, like what seasonal.
Do you want to?
Go ahead.
Thank you very much. I enjoyed both your talks today. Carsten, I do have a question for you. And again, it's not a particularly well-formulated question because it's not really my field. So I guess my understanding is that you're saying, well, stress, there's a real temporal difference. You're differentiating stress from harshness based on time, as well as potential a biochemical metric that you can measure. Am I right? Because surely there's also biochemical.
The main difference is how to get away with a bad situation. If you can get away with a bad situation by investing energy and fighting it or running away, then it's stress. If you cannot do that, you have to endure it, then it's harshness. And by this, typically it is related to time. Harshness is something that lasts long. You cannot And some animals, of course, escape the harshness by migrating, so they invest
Yes.
energy to migrate, so they don't have to live with the harshness. But yeah, I think it is in the nature of it that harshness is longer lasting, so by investing a lot of energy quickly, you can make it go away.
Okay, but I was thinking that they're both adaptive. You need to be able to adapt to stress as much as you need to adapt to. Yes.
Specific bad situations where you can overcome it by investing energy, and the harshness response is also adaptive by being able to endure it, to live long enough, get back into the good period, so you can reproduce and have productive success and evolution and fitness.
Okay, and how long do they live for? I don't think I.
Striped mice?
Yes.
So under natural conditions, they breed typically for only one season. The breeding season in South Africa, due to the rain, is very short. It's only about 3 months old, and in most years they cannot breed in the year they are born. So they enter the dry season as young adults, then they have to survive and also still to grow a little bit to be competitive. Then they enter the next breeding season, and then they reproduce, but nearly no one survives for the second year. In 25 years with 10,000 individuals, it was less than 1% that can make it the second year. It's not impossible, but basically 1 year of breeding. And it's depending on the year, it's between 1% and 60% who survive dry season. There's huge variation depending on the harshness, which means when you survive and breed, you already have higher reproductive success than the mean of the population, because the other half is already dead. So it's really very important evolutionary aspect. When you manage to survive, then you already have a higher fitness than most others.
So you have to survive your first dry season in order to reproduce. And then you don't tend to survive your second dry season.
The dry seasons, they differ dramatically from year to year. And the interesting thing is in this system, it means it differs dramatically from generation to generation. And whether your parents went through a harsh or not so harsh dry season does not at all predict the dry season you're going through. And we think that's also what led to the evolution of the flexibility. They cannot predict the conditions under which they grow up, the conditions under which they have to survive the dry season. So for us humans, it's unpredictable. We cannot predict it statistically, and I think the mice can't either.
Thank you.
Thank you for a fantastic talk. I enjoyed the difference between solitary and not. I had a question sort of arising from what both of you have said. What's the benefit of the kinship in this case? Is the relatedness higher or lower for solitary species? Of course, you had solitary in 2 different groups, but is it higher? Is the benefit greater for having a neighbor that's in kin? Is that greater benefit than if you have an immediate neighbor in a group? So if you actually measure the degree of kinship, is it higher in solitary than social groups?
Yeah, so for my species, it was much more higher. I would say I found it to be much more higher for the solitary.
Good, yeah.
Individuals. Whenever they did form the small family groups, it was always a special case. So it was only during periods of very high population density. Then you find this for small family groups that forming. But during normal time, then they were mostly solitary. So it seems to be driven by this high population density, where now the availability of lodges is restricted to the number of rats that are available. There's too many rats and not enough lodges available.
Right.
Whereas when there's now less individuals but more stick lodges available, then they can have just neighboring rats who are still related to them.
Unrelated question. Is the...
Yeah, no, could you speak into it?
Oh, yes, sorry. I couldn't help but notice that it looked like there might be a higher proportion of predaceous species in the solitary. Is that a coincidence, or is that not true? In other words, is there something about being predaceous that means you have to be spaced out rather than in tight groups? Although, of course, wolves and other canids are different, but is that too fast?
We think that In mammals, predation is very important for the evolution of group living, but that's mainly the case for large mammals, which can defend themselves against predators. If you look at a gazelle or buffalo, it's compared to a lion quite big. But a mouse or rat is compared to a jackal, to a bird of prey, small. So in other words, a group of buffalo might fend off a lion, a group of mice can never ever fight off a jackal. And interestingly, the striped mice, though they are group living, they're solitary foragers during the day. And we think that is the strategy to avoid predation. A group of mice running around would easily attract predators. So they go foraging alone and they might meet, but it's the sociality mainly occurs at the nest where they sleep together and warm each other. So that is the special case of the solitary forager that's group living.
I've got a very simple and curious question of curiosity. The little Karoo rat, you showed the bush hut that it lived in. Do they, in and under that, do they actually build any part of their nest, or are they just total opportunists?
So from a previous study that we, like last year, we had a student who was able to construct one of the lodges, and inside the structure itself, he did find some nest chambers in there. So we don't. What I still want to see during my postdoc is whether or not individuals within the lodge do they, if they share their nest chambers, because we do find maybe 2 or 3 nest chambers within a lodge. So even though it's just still the one big structure, and then we see individuals in multiple individuals who can share the structure, but are they also sharing the nest chamber? I'm not sure yet. So that's something to look into.
Thank you. It's a question for Carsten. I was curious when you said that each season could be slightly different, so the animals don't really know if it's going to be how short or how long that harshness is going to be. I was wondering, does it change them genetically? If they've had like a fairly good season, do they turn on or turn off different genes to make it for their children? Do their children actually change? You know what I mean? So, like if we...
So we don't know yet, but there's 2 things you mentioned. First would be epigenetic changes.
Yes.
But these you would, like parental effects, but these you would predict if the environment is predictable. But in this species, it is not. So in this species, I would really not expect any parental effects. Whether, because a mouse might grow up in a very great environment and have a lot of babies and have children, then go through the worst crisis since 20 years, and there might be epigenetically primed, there's a lot of food, a lot of water, but they really go through hell. And this can happen. When I started in South Africa, I had 2 very great years. There was a lot of mice, a lot of reproduction. I marked 200 mice. I was until January. I came back in August, and out of 200, 2 mice were left. Because it was the worst dry season for decades, it didn't rain at all. Then I looked at these 2 mice and 1 fall dead. Yeah. And then I had to increase the field size from 1 hectare to 10 hectare. All my research questions were gone, and to study them. So it was, for me, totally unpredictable. For them, it is also. That was at the very beginning, and unfortunately, did not have tissue samples there. But we are collecting our tissue samples through all the years, and if such a tragic event, such a severe drought would occur again, then we would have the samples now available to see whether there's changes in genotypes, in genetics, induced by one such terrible crisis. So this question we cannot answer yet. Epigenetics, I don't expect anything. Genetically, I would expect something, and maybe if something bad happens, we can answer this in some years, but I cannot predict when this might happen. But I plan this year to spend 1 month working on a research proposal on each of these.
Okay. All right. Thank you.
Hi, both great talks. I've got a question about the Karoo rats. So with the kin being potentially closer to each other, do you think that's an active choice by the animals, or do you think it's just a matter of they haven't been able to disperse as far?
So with the what I didn't show here, if I can show it with one of the pictures. So with all of these lodges, what I found was that when the population density is low, a female can occupy multiple lodges. So she can make use of maybe 2 or 3 lodges within her home range. And then when she has the first litter, they are in the prime spot to inherit those lodges that she's using. I'm still working on the paper. Hopefully it will be out sometime. But then, of course, it will be interesting to see. We know that the first litter is in a better position to inherit from her. But then what happens to the second and third litter is a different story. What I'm seeing now, because we have a very high population density, is that the third litter, because now there's no more lodges to get from mom and no more lodges available within the immediate vicinity, they tend to stay then with the mother. And then this is when we're seeing these small family groups occurring. But what Carsten was just whispering to my ear is that we have now a PhD. We're gonna have a PhD student with a new grant that we will hopefully get. And we also want to see not only the matrilineal relatedness of the individuals, but also the from the father side. So we see males roaming over large areas. So we want to see, are the females within, all the children or all the offspring within this specific area, are they all from the same father, or is it different fathers? Or
Yeah, very interesting. Thanks.
I also thank you guys for the talk. I've got 2 questions. One is probably a shorter one. When you mentioned that it's pointless to look for something that doesn't exist, as in the feeding activity goes down in the dry season for the striped rats, right? So as if the rats would know it's pointless. And I wanted to ask you maybe to go a bit more into this. Is there like learned behavior, or why do they know it's not worth it venturing out and looking for something, or like, especially because it's so unpredictable apparently, right? So just I'm wondering how this has established itself, that these rats don't expend the energy to venture out further. And the other one is harshness as a property of an environment. Like, I'm actually quite curious about this. We are here in the rainforest, right? And I see a lot of wildlife struggling at sometimes at a year. So I'm actually just wondering, what is the metric that compares environments with each other to say, okay, this is a harsher one than another one? So how happy do the animals have to be, or how unhappy, or how often do they have to be out of the comfort zone? So I found that, I mean, I don't know if there's a very quick answer to this, but yeah, just leave it there.
First question, I mean, these mice have home ranges of maybe half a hectare, and of course they know their home range very well, and they see the change that occurs continuously. So they know there's no food. There's no question about that. And they also know their surroundings. For example, in the you see it's along a dry riverbed, and it nearly never has water. We have something we call a shrub belt, and the more far away you get from the riverbed, there are less shrubs there are. But in the moist season, then there's ephemeral plants growing, especially wildflowers, rich in protein, rich in everything. And the mice are moving away from the shrub belt more outside into the barren areas in summer, and shift their home ranges to get better access to this food. So they know very well how much food there is in their home range and in their surrounding. Of course, they could have the idea that the grass is greener over this mountain, and if they have this idea, they're out of the gene pool. Yeah? So I mean, this is just natural selection. So a mouse would behave like this, and could be food somewhere else. It won't contribute to the fitness. For the second question, I'm not sure I can give a good answer, but what is important here to say is that harshness is not just a trait of the environment, but it depends on the species. For the same species, For different species in the same environment, it might be harsh or not, depending on what that species is feeding on what its life history is, and how big it is. So I don't think environment is harsh per se for everyone. Maybe in extreme cases it could be, but it also depends on the species. Yeah. Like for the bush carrots, the striped mouse occur in the same habitat, but the bush carrot is purely frugivorous animal. It only eats plant stuff. While the striped mice are omnivorous. They also eat seeds, which the bush carrots don't eat, but there's still seed banks also in the dry season. They also eat insects that are available. And we also have the elephant shrews there, which only eat insects. So the harshness of the trout is much worse for the bush carrots than for the striped mice, while for the elephant shrews, they always have access to insects. The harshness would start at a much later stage. So it's not only the environment, but also depends on the species specific life history.
Thank you. Pum. I have two questions actually, and the first one's probably more of a thought than a question. So the contrast between harshness and stress kind of reminds me of an anthropological observation that humans who live in pre-agricultural environments, like let's say hunter-gatherers in South Africa or Amazon tribes, they live in a much harsher, like objectively, environment, but they're happier. And also the observation, like especially from colonial era began, let's say, that as soon as colonialism came to them, like there was a spike in unhappiness. So like they felt kind of lost, and like it seemed that like they should get some benefits from it, but even if they got them, even if they got like homes, for example, more abundant food, they just like felt lost. Usually it's interpreted as like indirectly correlated, that basically harsh environment distracts them from let's say being stressed. But it seems like from like your presentation that there is actually a direct correlation, that like harshness directly reduces stress. Yeah, that's like more of a thought. And then like the question, I guess, like back to chronic stress in humans, like for example, people stuck at work that they don't like, or in marriages. Why doesn't stress in these instances converts, let's say, into harshness? Like for example, let's say I'm stuck in a marriage with a witch for a wife. Like it seems reasonable in this case to just save energy, to lay low, keep my head as low as possible, and preserve energy. Yeah. So does it mean that it's not so much the duration of the trigger, but like the nature itself, that like some triggers that are inherently stress-inducing and some triggers are hardship-inducing?
So all of this is very speculative, and I would prefer to discuss this over some beers. Decide later from what is my opinion. Of course, I have an opinion on this, but I want to strongly say this is an opinion, might be totally wrong. First, I do not believe that hunter-gatherers are per se happier than we are. For example, the murder rate in Khoisan Bushmen in Namibia was clearly higher than in Australia or Germany. If you talk about the Indians in Amazon rainforest, it's extremely much higher, and there's conflict between the sexes is very, very bad for the women in these tribes. So I'm not sure that they are happier per se. For the second question, I think there are cartoons saying that men develop the ability to listen to their wife without listening to it. That is a way to endure this, and then it's harshness and not stress. I don't know if this is true. And of course, we all have different mechanisms how to deal with our social stresses and they become long-term stresses or not. And of course, every relationship, doesn't mean if it's married or not, in social animals always has conflict in it. There's no living together without conflict. So that there's sometimes stress that is in the nature of being social. If it becomes too long-lasting, then it's, I would guess, detrimental. But then in our social species, there's also pressures from outside, from the family, from friends, and the benefit is something else. And last but not least, one thing to mention here very importantly is evolutionary fitness is very different from proximate well-being. Yeah, you might feel very bad and still be evolutionarily successful because evolution doesn't evolve us to feel good. It evolves us to do something when we feel bad, to have higher fitness, and it might still be evolutionary the best to stay at a well-paid job that sucks, and you have a lot of money and you can inherit it and you have a lot of children, and it might be very harsh for you, long-term stress, but evolutionary it might still be the best. Yeah.
Thank you very much.